All right, hello guys. We're going to go ahead and finish our notes through this video that I am creating for you. Yesterday we just finished talking about how to focus in your specimens and how you should always focus on scanning power first, then low, and then if you're able to with the slide that you're using, then you can go to high power. The rest of this kind of notes / instructions will concern our laboratory work. And so one of the things that we will be doing in our upcoming labs is we will be sketching or drawing out our specimens. So some helpful tips to help draw or sketch the specimens is to always use a pencil, just because nobody is perfect. And a pencil allows us to erase and shade different areas that if we were using a pen would probably not work out so well. In addition to this, all drawings should include clear and proper labels. And when you draw, you should be drawing things that are large enough to actually view the details of whatever you are looking at. And your drawing should be labeled with a specimen name and the magnification. Now with our labels, because as scientists, sometimes a picture doesn't tell us everything, which is why we label things. With our labels, your label should be written on the outside of the circle. These circles will be provided to you in your lab, so they're going to be built into your lab paper. And this circle indicates the viewing field as you see it through the eyepiece. So with your drawings, your specimen should be drawn to scale, meaning that if your specimen is going to take up the entire viewing field, your drawing should reflect that. Or if your specimen only takes up a quarter of your viewing field, your drawing should reflect that. Okay. So here is an example of what I would expect for your labs coming up. So the circle is obviously what you are seeing in the microscope. And this drawing shows me that they see this particular organism that has a cell membrane running along the outside of it. It also has a nucleus here in the middle. Notice the labels are on the outside of the cell. Also notice that we have a name, although it is spelled incorrectly. Amoeba is A-M-O-E-B-A. So please spell your specimen name correctly when you are drawing and writing in your lab stuff. And then it has this, the magnification. And so this 400x is the total magnification. And if you remember from yesterday, total magnification for high power is 400x. So this tells us what it looks like on high power. This would look very different if we were on a different objective or have a different magnification. With our labs, for several of our labs, we will be making what we call a wet mount slide. And to make a wet mount slide, you are going to gather a thin piece or thin slice of whatever specimen we're looking at the time. And we like to get a thin piece because if the specimen is too thick, then the cover slip that we're going to put on top of your specimen is going to like wobble on the top of your sample, almost like a seesaw or a teeter-totter. And with that specimen being too thick, then we're not going to be able to use high power with it. We'll also have trouble magnifying it and getting it in focus if our cover slip is wobbling on the top of it. Okay. With our wet mount slide, after we obtain a piece of whatever our specimen is, we're going to take a slide, and the slide is a rectangle shaped slide, and you'll see a picture of this on the next slide. And you are going to take 1 drop of water and put it on your specimen. So you're going to have your microscope slide, you're going to put your specimen on the whatever it is you're looking at on the slide. You're going to put 1 drop of water directly over it. And we only want to use the 1 drop of water because if we put too much water, then the cover slip will often float away. It can make it very hard to draw the specimen, because sometimes your specimens can even float away as well. In addition to this, too much water is just kind of messy and makes for a lab and a lot of cleanup with it. Okay. And then when we are putting on our cover slip, your cover slips are typically square, and you're going to place the cover slip at a 45-degree angle with one edge touching the water drop, and then gently let it go. When you do it correctly, this will allow the cover slip to perfectly fall over the specimen, and it almost makes like a little bit of a seal holding your specimen in place. So if you want to see picture view here, this is our microscope slide, which are glass rectangle shaped. Okay. Here they've already got the specimen on the slide, and they're going to put 1 drop of water, seen here, on top of their specimen. Then the next part, we're going to take a cover slip. Cover slips are very thin, square pieces of glass that we use to kind of secure the specimen. It also helps with bringing certain specimens into focus better. You're going to take that cover slip, you're going to put one edge on the slide, actually even touching the water, and then you're going to place your slide at a 45-degree angle and hold the tip of it with your fingers like you see here. And then you're just going to let it drop. Okay, now when it drops, it will form this seal between the water and your specimen. You never want to push down on this cover slip. One, the cover slip is super thin and you could even break the cover slip. But two, if you are looking at like microorganisms or other living specimens, if you push down on that cover slip, you can actually harm or kill whatever you might be looking at. Okay, so you literally just drop it and then you're done. Now, one of the next steps that you can do after you make a wet mount slide is you can actually stain it. So we stain specimens for a variety of reasons, but most of the time we stain them so that we can see certain pieces and parts. There are a whole bunch of different types of stains. There's iodine stain, which is like an orangey yellowishy color. There's methylene blue, which obviously is like a very almost turquoisey blue color. And there's greens and purples and pinks and all kinds of other colors too. So when we're working with stain, we always make a wet mount slide first. After we make that wet mount slide, then you're going to put 1 drop of stain on the edge of the cover slip. And when we're doing this, it seems a little weird that we're not dropping stain directly on the specimen. But with these specimens, if we put a drop of stain directly over what we're trying to look at, sometimes it's too dark. It's too much stain. So what we're going to do instead, we're going to put 1 on the edge of the cover slip. And then on the opposite side of the cover slip, we're going to take a piece of paper towel. And we're going to just push the paper towel on against the edge of the cover slip. So what happens is that paper towel draws the water out from under the cover slip, and the water, as it's moving, is actually going to help drag the stain underneath the slide. It's pretty cool to see, but that's how we're able to disperse the stain easily and be able to not stain our specimen too dark. Okay. Now, as soon as the stain has covered the area containing the specimen, you're finished. The stain, whichever stain you're using, does not need to cover the entire cover slip. If the stain does not cover as you need it to, you basically just repeat the process. You get a new piece of paper towel and you add more stain. Now, when we stain specimens, I will be helping you the first time around, just because it is a procedure that sometimes we struggle with as students until we get better practice at it. And then always, if you have any extra stain, whether it's on the slide or on the table or on the counter, just be sure to clean up after yourself, because stain is called stain because it stains things, and you don't want other people or yourself getting it on your clothes because it will not come out. And so what we see here in this picture, someone is creating a wet mount slide in this first picture. They're holding a cover slip at a 45-degree angle with a pair of forceps or tweezers. They're going to let it drop. And then in this picture, they're taking a drop of methylene blue stain and putting it on one side of the cover slip. And then they're taking a piece of paper towel on the opposite side. So notice the stain is on this left-hand side, and then the paper towel goes on the right-hand side. What happens is that paper towel draws out the water from the wet mount slide, and it draws that drop of stain across your specimen, staining it correctly so you can actually see structures instead of just a big dark spot of stain. And so when we are done with our labs, we will always clean up pretty much the same way. And we will go through this whenever we practice our hands-on microscope stuff too. But with our microscopes, we store microscopes with the scanning objective in place. If you remember from earlier in this note packet, the scanning objective has a red band around it. We always make sure that the dust covers are on them when they put them away, and that's just to keep them as clean and as nice condition as they can be. With you guys, you probably won't have to deal with this at all. Our microscopes are wireless microscopes, which means we charge them almost like you would charge your cell phone. They have cords that look like either laptop or cell phone chargers, and then you can unplug them and they hold that battery life. So more than likely you will not have to deal with it, but if the chargers are out, we'll make sure we put them away. And then the slides, so the rectangle glass microscope slides, we actually reuse those, so we wash them in the sink. Sometimes we have to use a little bit of Dawn dish soap to get the stain off of them if we were working with stain in that lab. So we wash them and dry them, and then we put them back in the slide boxes to be used later. And then the last part here, we throw our cover slips away. Now, the key point here is that we do not throw cover slips away in the regular trash can. That's because cover slips are glass, and throwing glass things away in trash cans are a big no-no because they can hurt people who collect the trash. They can hurt garbage men, they can hurt the animals from the landfill. And so with any glass, even if it's just a small amount, we always put it in a either glass collection container or we put it in a sharps container, and I will show you where that is when we start doing labs with cover slips. And this is going to go ahead and conclude our notes for today. And so keep in mind that you can go back and watch this video and pause it if you missed anything. And other than that, I'll see you in class.